Hello everybody, my name is Ben Harvey. So when we need to understand and interact with the world around us, timing of course is very important, as we all know. And humans have this great ability to combine our senses of timing from vision, from hearing, from touch, and this gives us one sort of unified temporal perception. And then we're able to make skilled movements that follow that timing. Okay. So such time perception for multisensory integration is most relevant for short events because most events in the world last under 1 second. So here we're going to focus on sub-second timing. But despite the central role of timing in perception, multisensory integration and action planning, the mechanisms of the temporal processing in the brain remain unclear, and that's why we're all here, of course. Following similarities in perceptual properties, we hypothesize that the processing of visual event timing may rely on similar mechanisms to visual space and number processing. That is to say, we were looking for timing selective responses organized in a network of topographic maps that hierarchically transform the representation of timing. And that's exactly what we see for visual space processing. We see a network of topographic maps in the early visual cortex, for example, where the preferred visual field position, in colors here, gradually changes across the cortical surface. In other areas of the brain, we find neurons that are selective for numerosity, that is the number of items in a display, and they parametrically change their responses as we change the number of items in the display. And here we see the numerosities in different colors that produce the largest response at each cortical location, and these again gradually change across the cortical surface so that the numerosity that produces the largest response at each location is kind of mapped onto the cortical surface again, forming a network of topographic maps. So we wanted to extend the idea of timing-tuned neural populations to 7 Tesla functional MRI. And so we did this by very gradually varying 2 parameters of the timing of these events. And the reason why nobody uses fMRI in timing, right, is that fMRI has terrible temporal resolution, right? But the trick here is that we very, very slowly change the timing. So we show lots of events of the same timing in a little period of time, and then we change to a new timing, right? So we changed a couple of parameters here. First, we changed the duration of the events. So here, for example, we always have 1 event per second, right? So the frequency of the events is always the same. But we're changing the duration gradually from 50 milliseconds up to 1 second. But we also changed the frequency of the events. Specifically, we changed the period between the events, right? So, the interval between the events, that is, from 50 milliseconds, very gradually changing up to 1 second between the events. And in this case, the duration was constant, so it was always just a 50 millisecond flash. And in a third condition, we varied the duration and the interval, the period, together, so that when one dot disappeared, another one appeared on the same frame, so the luminance of the display always remained constant. So this is the fastest timing, and this is the slowest. All right? So we gradually sweep from one end to the other. Okay, so the subject's task was simply to press a button whenever they saw a white dot. So they're not making any timing judgments here. All right, so it's a passive task, basically. But we need to make sure our subjects are looking in the right place and paying attention, of course. So we can see any repetitive event's timing as a point in a 2-dimensional space where one dimension is the event duration and the other is the period between events, the interval between the events. Okay. And here we can see the condition at the top where the interval is always 1 second, but we're gradually varying the duration. Here we can see where the duration is always 50 milliseconds, but we're gradually varying the interval. And here we can see where we're changing both. Now, we then want to fit a 2-dimensional Gaussian tuning function to describe the responses to all of these different stimulus configurations using just one model. Okay. And so we added a 4th set of timings, a 4th kind of stimulus condition, where we just kind of sample some timings in the middle of this space to better constrain our response function. Okay. Now, the nature of the resulting response function can tell us a lot. Right? So what we see here is sort of maximum response where the interval is 800 milliseconds and the period is 200 milliseconds, so there's really no relationship between those. Okay. But of course, it could be that the interval that produces the largest response and the duration that produces that response are strongly correlated to each other. So here we can see this is kind of on the diagonal. It could be that the tuning function is elongated in one dimension more than in the other. So this, for example, would be relatively fine selectivity for duration and relatively coarse selectivity for interval. Or it could be that the tuning function is angulated so that the event duration that produces the largest response will change as we change the event period. All right? But of course, there's still a maximum response for a particular combination of duration and period. Okay. So to analyze our data, we use an extension of the population receptive field modeling approach to fit these tuning functions for event timing. So we start with a large set of candidate tuning functions that might look something like this, for one example. Okay. And if we know all of the timings that we showed in all of these different stimulus configurations that we're looking at, we can kind of take slices through this tuning function to basically tell us at every timing that we show what neural response amplitude our model would predict. Okay, so this is what we see here, is basically these cuts through this tuning function here. And then we also know when we showed, specifically when each event ended. So these dots appearing and disappearing, we kind of fix our responses to when each event ended. Okay, and we can see that this changes. All right, so it's not that this is the timing is regular. Sometimes the events are very frequent, right, which would say that we're likely to have a large response to those more frequent events. And sometimes the events are less frequent. All right. And so if we look at the interaction between these kind of slices through the tuning function and when each of these events happened, this basically gives us a prediction of the neural response amplitude that we would expect to see if this tuning function was responding to each of these stimulus conditions. Okay. Now, because we're changing the frequency of the events, we might not assume that the response amplitude linearly scales with frequency. Okay, so it's well known from many branches of neuroscience that the response amplitude to repeating events decreases as the frequency of the events increases. So basically, if you have one event per second and you have 10 events per second, the response to 10 events per second is not 10 times as strong as the response to one event per second. And so we can summarize that using a compressive exponent on the response amplitude, so that basically the larger the responses get, the more they're kind of suppressed, the more frequent the responses get, the more they're suppressed by previous responses. And basically this is the trick here, right? So first of all, we're changing our timings very slowly, right? And second, we're modeling how the response amplitude accumulates with event timing explicitly as part of our model, right? Which allows you to investigate timing using functional MRI's terrible temporal resolution. Okay, so then we can get this prediction of neural response amplitude, which is kind of the combination of these two things. And we can convolve that with a hemodynamic response function to give us a prediction of the fMRI response amplitude we would expect to see if this neural model was responding to these stimulus timings. Okay, and then we can compare that to the actual recorded data from every recording site and basically determine the correlation between those two, the prediction and the actual data. And we can do that for every possible neural tuning function, and indeed every possible combination of preferred duration, preferred interval, the tuning width on the long axis of this function this way, the tuning width on the short axis this way, the angulation of the function, and also this compressive exponent. So we test all possible combinations of that large set of parameters, all right? And then we see which one of those for every MRI voxel best predicts the response amplitude, the response time course that we measure. Okay. So this is all quite complicated, but basically what it does is it gives us it turns our fMRI time course data, right, into a tuning function that can be summarized by a small set of parameters. Okay, so so far I've kind of told this story as if the only possibility is that our tuning function is selective for event duration and interval. But that may not be the case. It may be that the tuning function is better parameterized another way. So, for example, it might be that the tuning function has one dimension that's selective to the event duration and another dimension that's selective for the time between events. Right? So that's another possibility. Or it could be that one of our events is the one of our axes is the event interval, just like here, and the other one is how full that event interval is. Right? And we can fit tuning functions in all of those different situations and see which one best predicts our data. Right? Now, because these different spaces are just mathematical transformations of each other, right, this is specifically for a parametric tuning function. Right? If you have a non-parametric tuning function, this can also just be transformed between these different spaces. Right? So this is specific for parametric models of the responses. But we need parametric models because those parameters allow us to make predictions of what we see in our fMRI response time course. Okay, so we can then compare lots of different candidate models that each give a different story of what the timing selectivity is. So, for example, in our worst model, our neural populations are just responding to the temporal frequency of the events. Okay? And temporal frequency doesn't give a different response for every event. It's just an analysis of the waveform of the luminance in the display. But if you make it so that you see a specific response per event, which is indeed what we see, the goodness of fit goes up a good bit. If we allow a tuned response, or a Gaussian curve, our tuning function gets yet better. And if we allow a two-dimensional tuned response, and specifically one that also includes this compressive exponent, okay, then we see the best fits, and this fits better than these other candidate models for things like the occupancy of the event or the time that the event is off, as well as the time that the event is on. Okay. So we test lots of different possibilities, and this makes us confident then that our neural populations are indeed tuned for the duration of the events and the interval between the events. Okay. So we can see several brain areas where these models capture the data really well, and these extend from the lateral occipital cortex through the superior parietal lobule and into the prefrontal cortex here. If we compare the goodness of fit between the left and right hemispheres, the light bars here are the left hemisphere and the dark bars are the right hemisphere, we can see that the left hemisphere goodness of fit is always a little bit better than the right hemisphere for the same subject in the same area. And we can also see that the maps are a little bit larger in the left hemisphere than in the right hemisphere. So this tells us basically that these timing selective responses are moderately lateralized towards the left hemisphere. Okay. If we then project the duration and the period preferences, those are two of the parameters of our model, onto these cortical surface, we can see that in each of these areas there's actually a range of duration and period preferences. Okay. And we can see that the duration and period preferences within each of these maps is very strongly correlated. Okay. We can also analyze this range of duration and period preferences in different ways. So first of all, we can take the mean preferred duration, and in each of these maps, despite very different types of responses, the mean preferred duration seems to hover around 500 milliseconds. That is the middle of the range that we present. And the preferred period is just a little bit longer than that, so it's always just a bit above 500 milliseconds. If we look at the range of duration and period preferences within each of these maps, in each of these areas, we can see that as we go from the back of the brain, the lateral occipital cortex, to the front of the brain, the premotor cortex, we see a gradual decrease in the range of both duration preferences and period preferences of our recording sites. Okay. And we can also see that within each of these areas, there's a gradual progression of preferred duration and preferred period across the cortical surface. So we're seeing topographic maps of timing. Okay. So those are some of the parameters, right? That's just the preferred duration and the preferred period. But as I described in the modeling, there are a lot of parameters to these models to really make it fit well. Another one is the extent of the tuning function. So our tuning function has a long axis and a short axis, right? And both the long axis and the short axis tend to get longer as the preferred duration of our recording sites increases, okay, in the posterior maps. So this is in the lateral occipital cortex. But as we move to more anterior maps, we see that rather than this sort of linear increase with preferred duration, we start seeing another trend on top of that as well. So this V shape, right, where the tuning function gets narrowest in the middle of the presented range, so around 500 milliseconds. Okay. So this suggests that the presented range really seems to matter to our neural responses once we get further forward. And indeed, as we get to the frontal cortex, we no longer see this linear trend in the long axis. We really just see this V shape. Right? But if we ignore this change within each of the maps, we can see that the tuning function's average extent gets smaller and smaller as we go from the back of the brain to the front. Okay. We can also look at the tuning function shape. So as we go from the back of the brain to the front, again, we can see that the tuning function gets less elongated, so it sort of gets more rounded. But in all of these areas, it seems to hover around 45 degrees. Okay? So that basically means that we have an angulated response function, so that the response amplitude maximizes at a different duration for every different interval that we show, for every different period. Okay. The last parameter is this compressive exponent. So with a compressive exponent of 1, we see a linear buildup of response amplitude with event frequency. But with a lower compressive exponent, we see that there's a very subadditive increase in response amplitude. And this compressive exponent also changes between these maps from the back of the brain to the front, so that we go from basically a distinct response to every event, okay, at the back of the brain, to somewhat integrated responses to different events. So if I show you 10 events or 1 event of the same timing, you'll see the same response amplitude, basically. Okay, so we've seen a lot of these transformations, and these transformations of the response selectivity throughout the brain, and these tend to go, first of all, from the back of the brain to the front, and second, up the pre-central and post-central sulci. So we see a sort of progression in this direction as well. Okay. So all of these experiments are for visual event timing specifically, all right? But where they end up is in just next to the the somatosensory cortex. Okay, we see one map right next to the face area of the somatosensory cortex, a very sensitive part of our body. Another next to the hand area, and another next to the kind of arm and neck area at the top of the somatosensory cortex. And in the motor cortex, we see one map right next to the representation of our tongue and lips, and another next to the representation of our hand and arm. So again, these are areas where we perform very precise timing. Okay. So these are responses that we see to visual stimuli. If we do the same experiment, but we don't use visual events of dots appearing and disappearing, but we use auditory bursts of white noise with the same progressions of timing, okay, we don't see this network. We see a very different set of areas becomes activated. Okay, so this is the visual timing, this is the auditory timing. Right? So we go from really this network that looks like the frontoparietal attention network, basically, to this area here, really focused on the near the auditory cortex, and also this very repeatable area that we keep seeing, which seems to be next to the representation of the neck, which may allow us to orient to sounds very easily. Okay. But if we look at this area, we can see this has a particular shape. We can also do tonotopic mapping to localize the primary auditory cortex, and we can see that this area surrounds the primary auditory cortex. The primary auditory cortex is kind of the center. So here it really looks like there are responses in the auditory belt and parabelt of the auditory cortex. Okay. So if we look at these duration selective responses and compare them to numerosity preferences in the same areas, we can see that there's a lot of overlap between these areas, but there are some distinctions as well. Okay. So to conclude, we see an extensive neural tuning for visual event timing in the human brain using functional MRI. It's tricky, but you can do it. And so this is in areas involved in motion processing, like area MT, attention control, the superior parietal lobule, multisensory integration, post-central sulcus, and action planning in the premotor cortex. We see very strongly correlated and interacting tuning for duration and period or interval. So this suggests these are estimated by shared computations. And we see a progression in tuning widths from posterior maps where we see an increase with preferred duration to anterior maps where we see really a decrease in the middle of the presented range. So they seem to be focusing on the middle of the presented range in the frontal cortex. We also see a hierarchy from posterior to anterior maps where we see increasingly small, less elongated tuning functions. We see an increasing focus on the middle of the presented range, increasingly sub-additive compression of response amplitude. We also see that this is specific to vision. So if we do auditory stimuli, we see that responses to auditory timing seem to really focus on the auditory cortex belt area. And we also see that this largely overlaps with the numerosity map network that we've recently described, which suggests a kind of generalized quantity processing for visual quantities. So finally, I'd like to thank my collaborators on this project, Serge Moulin, Alessio Fracasso, and Jacob Paul, our funding agencies. If you're interested, there's a preprint of this already available on Current Biology's website. And if you're interested in a postdoc position, I'm currently hiring a postdoc. The deadline's coming up for that. Okay, so I'd like to thank you for your attention. I'll be happy to answer any questions.